In our next section, we will review the discovery timeline of the various components of the endocannabinoid system. Although cannabis has been used medicinally for over 4,000 years, the ability to identify its active components and discover how they interact with the human body has been a long and difficult process. The active molecules of the cannabis plant are completely insoluble in water, which makes them incredibly difficult to extract and identify. While researchers in the United States were the first to extract CBD and THC successfully in the early 1940s, it wasn't until 1964 that Raphael Mechoulam's lab in Israel was able to accurately identify the structure of both THC and CBD from the cannabis plant. From there, scientists used a radio-labeled synthetic cannabinoid to identify and map the first endogenous cannabinoid receptor found within the human body, which was named cannabinoid receptor 1, or CB1. The subsequent effort to identify and clone this first cannabinoid receptor demonstrated that it is one of the most abundant proteins in the brain, which has made it a very attractive target for novel drug development over the past several decades. It then wasn't until the 1990s that researchers made additional discoveries essential for the establishment of the cannabinoid research field. By the end of the decade, scientists had discovered two distinct cannabinoid receptors in the human body, isolated the endogenous cannabinoids found within the body, synthesized cannabinoid receptor ligands, and generated cannabinoid receptor knockout mice. The cloning of cannabinoid receptors opened a door to the development of synthetic molecules that can act as agonists and antagonists at the receptor level, which then allowed researchers to begin characterizing the role of these neuronal G-protein-coupled cannabinoid receptors. The development of knockout mice without a CB1 receptor, making them immune to the effects of THC, further proved the link between these receptors and cannabinoid activity.